Now they are indirectly coded through the expression or encoded through the expression of glycosyltransferases, which they have to be positioned in a certain way in the molecule so that you can get the sequences, specific sequences. Now, most of people will say, so what? But the whole evolution have actually preserved all of the glycans. And then we can see just a schematic presentation of the cell. And if you think about the multicellular organism, so every cell surface, extracellular matrix are highly glycosylated molecules, often over 50% of the glycan structure, which are also very specific for the cell type or even in the development stages and are expressed on every surface. So if you think about multicellular organisms now, what Eric was saying, now we shift to the other point of how does the cell, like this one, which has what we call the cellular code, Glycocalyx code, recognize where it is. It has to communicate, bind to something or not bind to anything. And what are the most peripheral molecules? These are the glycan molecules, right? So what we are interested in, what we will try to present here is really how these glycans, they have to recognize that they are each organism, they have to recognize self. And why we say that there are other molecules, proteins, and so which we recognize. But they are the most superficial part. So whatever comes first, it's your first sensor. Your area of your cell, which all of the cells have these glycans, right? So anything coming first to the cell, any signal, any cell with the other molecule, any hormone, any virus, any bacteria, anything has to first see that or touch it. And now the big enigma is for us, how does actually anything goes through this glycocalyx, what we call it. It's a cellular code. And how does this glycans work? So really what I will be talking is would be it could be the self-recognition, maybe it is the glycans, and how this cell layer, does it have any structure? Because we talked about morphogenesis on the cellular level, but there is a morphogenesis maybe on molecular level. And so this is the other part, and then actually what I will try to introduce was from my PhD studies about that there is a possibility that glycan interactions are very important because they are also close to the factor. And the difference in structure is always think about that the interior part of the cell does not have much glycosylation. It has monosaccharide, maybe some of the molecules, the secondary post-translational modification. But the cell surface and extracellular matrix and glycocalyx in general, and interior tissue on the apical side, are heavily. So, what is the second part of the talk when we want to describe it is to connect the structure of these glycans to their function. And so, this is myself presenting that, and I'm now at the moment with Eric's laboratory, with my wife also who is present here, and we are trying actually to connect the structure function using the different imaging assays, structural analysis by sequencing, by X-ray diffraction, and as well as a functional analysis in the different cellular assays. So basically, what we can see from this is that every, what we have heard here, that every multicellular organism is a unicellular organism. It's a very complex living system. You have so many different molecules, so many different cells. Tens of trillions of the cells in the multicellular organism, the larger ones, billions of molecules, which may be 100,000 or 20,000, depending on the cells are different. So just recognizing in general recognition is very important on the molecular level, which will then guide further processes. And so we are really interested in this mechanism, how the self-recognition and the molecular self-assembly will occur. And now we can obviously know all of that at the different levels. You can organism level, you can tissue, cellular, molecular
our goal is to drive these scales on each sort of scale. And so what are the molecules, just to repeat this because it's very important, people who study the membranes and the cells, they are very interested in it. It's very important because that is really how we get a cell to live and to survive. Then we have obviously the proteins, we have nucleic acid, which code that, and then we have the glycans, which are actually not important in the gene expression. So now, what is happening? The two cells are meeting, as they say. So, what is the first thing that should happen? We hear about multi-cell adhesion molecule, how they do it. But again, the glycans are most peripheral. This is kind of a hypothesis, and I think we will show you that it is with some imaging techniques actually true. So, two cells come together, right? Whatever is the cell, whatever is developmental stage or anything. So, the glycan, you cannot avoid the glycans because they are the first encounters of any cellular environment. So, somehow they have to possess this recognition function, which is the initial cell. And we have to compare this to the protein-protein and protein-glycan interaction, where the proteins are usually called molecules. And they're very important in many immunological cancer cell adhesion and beyond development. They're very widely studied. And as well as the protein-protein interaction. But we will focus on this glycan as the initial step of the cellular interaction. I will just briefly show you this experiment which actually we repeated when it was done about 150 years ago in Woods Hole, where Eric was working and I was working for many years and told me as well, and this is a fantastic place. And Wilson, he was one of the first people saying, how do the multicellular organism, and this is funky, so these are the cells of the first one is our how do they regularize this up? What is kind of image? Because we have to, by definition. So, what we have done actually was one of the people that worked there for a long time. So, what we have really is this we have three different species of form. So, what these people discover very simple things. So, okay, they can squeeze and they go in the single cell. If you just a concept, the health is very important for many different roles. And then it sounds to be so technical. You have a concept back, the other take the viral software. So there is a recognition happening there. And now the question of one of the molecules. So I think the most of the countries were the two people who in the past 4 decades were trying to isolate a small group, and we were the ones who went more to characterization. And what we find is that this is a new class of the so-called, we call them now bottle brush molecules influenced by Eric, the other Eric because you can see that in the moment of the next slides, they are large protein pores, maybe 500 nanometers or something, and with extended glycans like proteoglycans. But these molecules are not proteoglycans, and I will present you the evidence about that, and we call them a glyconectins. And so, for myself, working in Max Berger's laboratory as a PhD student, actually that's work was initiated, so many, many years ago. So what we actually found is the following thing, that for this same species, we call it glyconetic one because it connects cells like glycogen. So make this work. Everybody said so we have isolated this glycan molecules, this bottle brush molecules that they have general basis, and you can see G1, G2, G3. But this is kind of it's multiple bias purified. As a purified converted some proteoglycan plants. So, this is kind of the rate of the molecular weights and the sequences of the nowadays we get sort of the two main possible types of so they're quite well defined molecules. So what they do now, if we just take this purified glycans in glycocalyx molecules, and then we actually that was not very was doing this experiment and we found the most exciting aspect. We all collect, we put in each of these molecules of the magnetic state, we will not identify. You can find most of the way, right? And then we go to the different color beads with each of these purified glycocalyx. We add a calcium, and then we have surface interaction which lights up very specifically like glycocalyx. So that means any mix without negative like itself. So that means we have a molecular homophilic self-replication of these things which are actually glycan interactions, which are very calcium dependent, they get to the plasma where there is a tiny more calcium, which is a natural signal of calcium, which the sponsors of various countries are living in. Okay, so now the very important is that you self and then suddenly then quarterly manipulated and actually published in the Google Journal Science Innovation. It was accepted, it was important. We are very grateful for the other people also accepted. So what we have here is we have a combination of the different colors, labels that correspond to the colors. So we have like making one sponge cells and then like repeats, and then you see homophilic interactions. They are just like one organism. So they actually will incorporate a little bit. So, and then they kind of will actually sort out in a very short time. So we have on the cellular and molecular level showing that apparently these glycan interactions are responsible for self-recognition, which is very important for the primordial kind of immune system of how we recognize. So the other question is now, how does it translate in the other system? And we think that actually this is again one general principle that would be the first step. But the experiments there are much more difficult, and we can talk about it. Anyway, so once more to show now, this is now the full connective molecule with the purified, and you can see that the molecular masses of the really large. It's very virus, and then if you have the general for this, they together is that's the experiment of the life cannabis. That's why you do glyco blocking of the polyps that you can show the specificity of interaction. So now this is the picture which we try to take each of these molecules and then fragmentate the molecules in order to get the sequence. And that was one of the most difficult things to do because what we have found out is that these molecules are similar to protoglycan glycosaminoglycans, but their sequences, as I will show you in a moment, are very different from normal sequences of the glycans, but they have a similarity. They are very large and they're patterned sequences. They can be sharp, they can be rapid. But maybe the more individuals which I give that I first of all, I will show also that these antibodies are very specific. But also very involved, like oxidases, which would have they can assist in nature, but we never get finally able to try to hide the I don't want they get it. So we went out to the chemical approach, which is difficult to hydrolyze mild hydrolysis of this. And what we have obtained is these are the structures. I mean, if you are not a glycobiologist or so, I think this is maybe an interesting, but for you, but what you can see that there is a sequences, and always you can see that there is either in this case animals, equals either animals, equals, and there is always equals and equals within the chain. It's actually not seen before, of the long longer chain. And when you see another important feature is that even with boundaries, even there, that don't have molecules in some favorite part or period related to, and you can consider in the also the uronic acid, glucuronic acid.
So how we actually did this, it is obviously very long subject. Many years to come to this, 10 years. So you get some of the sequences between animal and mass spectrometry. And people who do mass spectrometry understand that in animal the glycans are the most difficult to do. Okay, so these are these ones more to show this sequences and where the calcium defines. We just taking one of them, but making one here in the ceramic show chemically, and this was not a simulation in another group. They show where the calcium and bind this molecular thing, and then how we can bridge this molecule. The piece you will have this homophilic glycan interaction. So now the calcium dependency is one very important thing, and it is specific for calcium. Magnesium cannot do that. So that means the pockets that you saw before are actually the pockets where the calcium, as a bigger than magnesium, can fit well and bridge the charges. So it is species specific, what we have shown before, and now just to give you the scale, because it's one function parts, the structural parts. These are molecules in very different stuff, very basically about parts. So if you now make significant growing, then what we do is that we show that this chemical is between 30 and 100 times the one particle chain. Depending on what you make it. And there are some intermediate secrets, but significant I didn't show them. So if you know the cluster rate, this is a really scale, and we can complicate with. I published that what you can see is that I will use the mouse if I can, yeah. So what you can see, this is the real size of the plasma membrane, and then this is like a transmembrane protein. This is like oscillation of the N and O-link glycans, and then you enlarge it, it looks like that very heavily glycosylated. And this is the size. If it will be just elongated, it's not elongated, obviously. So it is shorter than that in the real size. But these are the dimensions which are incredibly different. And what we are trying to do that on a little bit of the molecular level is that what we can see is that, like EM level, you would see these glycans which are stained with thin red. This would be AFM picture, which we did showing actually, possibly this should be the glycan chains on the circular protein core. And then if you compare that in the same scale with the M-link glycans, which are yellow here on the protein, and then if you will put our glycans repeating, this line will end up around this row, just to get the perspective of what is the cell surface. And now I will come to the structural part. How do we study how this glycan comes? Does it have any form? Does it have any structure? Is it organized? What are the pores that are there? Any pores? How anything can come there besides the recognition of the stuff? So since maybe 50 years, the people have been doing different types even of the cryopreservation, but with high dehydration or fixation of the molecule. And what happens there? It is what actually Emanuela was drawing, that was part of her PhD thesis. When you think about these glycans with these charges, these are so heavily hydrated. They're probably 10 times, if not more, hydrated than anything inside of the cell. And they're autophagocytic. So if you do any fixation and dehydration, or even cryopreservation, then you have to kind of use the cryos SEM, scanning electron microscopy. What you get? You lose, the water goes up. Now, these large molecules, which I show you, they will just collapse. I mean, it's obvious physical chemistry. And all the ions will accumulate. So what you get is usually the structures which then is kind of meshy, unorganized, which was, you think I should show it there, right? It's, okay. So this was the picture for probably 50 years. Then what we did is
with the help of Erik Adler and my wife Emanuela, we actually had an idea to use the cryopreservation without dehydration. So the minimal that, so what you do is you prepare, and she developed a kind of a procedure for that, that you do the high pressure freezing with the liquid nitrogen. Is a machine which costs several $100,000 just to do that. But you put, and she has a poster, so you will see that how this is exactly that. So you have this rapid freezing, you have the purification, so no change. It's really a snapshot of what it is, the structure. Then you, and this is in a capsule, okay? So then all is done out of the liquid nitrogen, and you burn a lot of fingers, right? So you have to put that into the other machine, which will now fracture this. So it will fracture at the place where is the minimal force. So it's not cutting, it is fracturing. So by fracturing, you will get a surface, and then you're still in the cryopreservation, no sublimation, no condensation, which you can control by the pressure and temperature. What you are getting is then incredible things. These are now the cells here. So these are the most native state that has ever been seen, okay? There is no fixation, nothing, inside and outside of the cell. So what you can see, it's kind of what we call organized array, and we call them a lamellar structure, which is probably not the best thing because people consider lamella something different. And we don't know still the 3D. But the coupled surface is very clear. So the higher magnification, you can practically see individual molecules of kind of, and we don't know in this case, these are the cells which are cultured in the carrier which you developed them in, and then image just like that. And then you can see, so you don't touch the cell, you don't do anything. And then you can see this very well organized structure. These particular cells, they express leucis and proteoglycans, and also what I think we have shown, they also express some of the glycocalyx. So we cannot yet identify which part of this, but this seems to be what we will hypothesize, the protein cores, and maybe with the lateral glycan chain interaction or glycan protein ones. So they get organized, this bottle brush in a line, and they make this. Now the question is, if you take more water, but still the cryo, right, that is, you just dehydrate this by what you call etching, then you will get kind of mesh for the acidity. So obviously, because everything collapses, so you move the water, things cannot enter the iron cell. So these are another cell types, which you can see this kind of array of this, let's call them now lamella, and this is the cell. You can see part of the nuclei. You can see wonderful structures also within the cell. You see that the cell is surrounded in a culture with incredibly large layer, what we tried to show before, of the glycocalyx. And although the all lamella appear to be similar, which they should, because these are all bottle brush molecules, they still have a very distinct features. And I think now we are trying to do the statistical analysis and measurement, which is very tedious and not easy to do. But we are getting to that. So there is a specificity on one hand, and there is universal kind of structure. So let's say, how does our sponge glyconectin do? So what we do here is now the purified molecule, which you see on the gel, we added the calcium, it gelates, we do the same thing, and you see this again, very similar lamellar structures, single molecule. This is the AFM image of the spread molecule. Now this is the glyconectin 2, not the glyconectin 1, which is circular. And apparently these arms are carrying the glycans, and this is the protein backbone. If you etch a little bit more, you can start to see the individual molecules even in the cryo, which are very similar to the spread. This is a non-native one that will show the spread model. And they should be more or less kind of very not collapse in the aqueous medium. All right, so now it is a model which is based on our sequencing, how it will look like. And then this is now schematic drawing of the yellow, the protein core of the lamella in the glyconectin. And here is based on our sequences and monoclonal antibody binding, fragmentation, all quantification. We have the sequences. These are the sequences that that are presented in a long polymeric chain, and they're like a zipper. You don't like the zipper, right? But I think this is the easiest for people who are not in the field, would close and with the presence of calcium and actually make this lamellar structure. So they would self-assemble it, and this is now based on the sequencing. We have partial sequences. There is a little branch which is now connecting the negative charges of the pyruvate, which we say sequence, and this is how we envisage the model from the sort of atomic, molecular, supramolecular level and to the cellular level, how this recognition by a glyconektins is occurring on one hand, and how they organize so far not yet before known ultrastructure, the native ultrastructure of the glycocalyx. So now the obvious question is, so what do you do if you remove the calcium? Because the gel will dissolve. So what you see here is after the treatment, this is a very short treatment, the lamellas and the organization is kind of getting loose. And if you do a little bit more, you can start apparently to see the individual molecules of the glyconectin, which again resembles a bit what we see in AFM. But this is a fracture surface which you see not 3D measures, just parts of the model. So, okay, so this is once more to just show you how it works, and then we have this fracture plane, and then if you look how much water is sublimated from the cross-sectional from the planar part, if you remove more and more water, obviously this molecule will collapse and make the picture that we have seen. So let me now switch to the fractional maybe part or measurements, which is a quantitative one, which is atomic force microscopy measurements between of binding forces between individual molecules. We link one glyconectin molecule to the tip, which was isolated with self-assembly lipid layer, and then we do the same on the mica, so we don't have ionic interaction which are long distance, okay? So when we do the approach on the AFM, when you do the approach on AFM, these molecules will touch and you read this so-called force distance curve because the lever is like a spring, and then you start to pull, and then you can see now the multiple peaks coming, and then there will be, this is practically the force that we measured. And what it does show this picture, several important things. What it shows is, first of all, the total energy of binding, the different distance where the molecules are separated. These are polymers, these are large. And this is incredibly interesting, what we did now after maybe 20 years or 50 years when we did the first experiments, the analysis of even statistical of this distribution of the peaks. And still being afraid to say somehow it matches the size of the glycan molecules or of the atoms. Okay? So, and I will show you at the end the table of the kinetics and everything, what we sort of are now calculating. So, what is the functional meaning of that? Which initially Octavian and I did this, was the functional meaning of this, that the single molecule of these augmented atoms can hold weight of 1,600 cells. That was the first ever explanation, the single molecular level, that the cell adhesion molecule, in this case glycan-glycan interaction, can actually support the anatomic integrity of the body, but are at the same time recognition molecules, which make the organized structure of the glycocalyx. And then we ask ourselves, which I will come once more after this, how anything can come through this. And we have a lot of discussion, and we can talk about that physically. It means something has to go through. When I asked Trudy about the recognition, so something has to go sequentially through that to compete with this binding. And this is really, I mean, kind of a complex thing. So whatever it does, even the drug, even the monoclonal antibody, anything, most of the cells will have a large glycocalyx, few hundred nanometers. Other cells have millimeters and so on. Anyway, so this is a very challenging thing. So let me just show you these three molecules. And what we did here was originally is that you see the image of the molecule. These are bottle brush. We have now better image, but I didn't put it in the right time. So these are bottle brush, and then we do the binding of this molecule to itself, binding to non-self GM2, GM3, and then you see actually they do bind to each other. And the force of this binding is actually at the level of of the selectins binding, the lectin binding to the glycan. So it's not zero. And you can see even in adhesion, they come and then they actually sort of go to equilibrium and this. So nothing is a zero binding. That was our conclusion. Everything, if they are not repulsive, everything binds with a certain affinity. And we have to keep that in mind for the drugs, for antibodies, for anything. In immunology, this is now quite well understood. So anyway, you get this again diagonal, which you show that there is a specificity which can drive this, what we have said, self-recognition and adhesion. So, so the take-home sort of parts is the glycan to glycan is cell recognition adhesion in peripheral, glyconectics and bottle brush glycopolymers. They are species specific. Most important, they are highly polyvalent. Without that, you cannot actually do anything. The polyvalency is very important. When the affinity of the single site, I will show them again in the next slide, is actually below the hydrogen bond and below the KT. It's like, or just at that level. So you would say the individual epitope will actually do nothing. But once you get the comparativity, the whole thing is zip. I like the word zip. So, and this is now a single type of the site, which are not very different, but still they are sufficient to make this recognition in the polymeric way. And so what is the point that they can provide high affinity, specificity, but reversibility. What if you can see, and I think this is the kind of take over. So this is a schematic of this bottle brush again to show, to compare with that. And then we have these three, these are pyruvylated and sulfated and carboxylated more in the GM3. So they have other groups as well, hyaluronic acid. You have a backbone as I showed in the other one, and the glycan chains. Now, just to keep this number, this is now just a very, from the AFM data, just a short summary of that. So if you, what we measure is the hierarchy of this interaction in the environment, that they live. And that's how we did the AFM experiments, right? When we measured that. So you just take a single connecting molecule, 10 polysaccharide chain, and maybe 30 to 50 epitopes. So you can see you get to from 500 piconewtons, which I showed you can hold the cells very well. You get per glycan chain 50 piconewtons. These are the multiples probably that we are seeing in our AFM experiments. And then if you go per epitope, you get 0.5 piconewtons. So in the femtonewton range, which is about in the KT range. Now, these are all data now derived from the AFM, which I did lately, and we still are working on that. So if you think now about the binding of the calcium and the different molarity, so you can see depending on the sulfated, pyruvate, or carboxylated, they have different affinities for the calcium, just the calcium binding. And then they get usually saturated at around the 10 for this type of the molecular number of the cell, or 10 millimole. So now this is the very interesting table. And then, so if you now look at GN1, GN2, and GN3, we have also the GN4, but for the simplicity, let's put all this. So force per chain, force per 550, what they say, we just put them kind of similar for the homotypic binding. They are a little bit different. This is just to simplify it. So what is the force for epitope? Piconewton 0.5 to 1.7. So we put them there. And then epitope lifetime. What would be the epitope lifetime to live? It is in microseconds, okay? But this is because of the affinity. Also, the calcium plays a role. It's quite a big difference, which is interesting. These are the KD constants derived from different types of the experiments. And then we have... this is the calcium binding, which we have shown K on in molar per second. So it is 10 on the incredibly, I mean 10 on the 8, right? I think is that. And then the number of the collision, you have again what we can calculate per 10 per cells per milliliter acid, and it fits with our experimental data. So we can transfer this AFM measurement connect with our reaggregation or bead study. So it fits very nicely into that. So the number of collision, and then you have how stable will this. So the three chains, zip chains will be actually this reaggregate may stay stable, and what we prove in the beginning with the FF, so that the sponge can survive all of these things which are happening in nature. So it is indeed preserving anatomical integrity. Anyway, so these are showing here that what is it, the severe storm, you can calculate the wave forces in that. So now these are the reaction time to reach a certain stage after the reaggregation. And again, this is coincided with the results, not to bore you with the details of the aggregation study, which many people did beside us. Now, I just want to say to many different people, but these are the major people who really presented most of that. There were many other students for sequencing or so on. But these are the key people who developed the ideas and who executed these ideas in the results and experiments. This is Octavian, the colleague with whom I did most of the work. Eric, with whom we work since 10 or more years, with his incredible help and knowledge in the physics and polymer science, which we were lacking very much. He used to contribute. This is Emanuela, actually my wife, who is working with me, with mainly now with the last time with the cryo experiments. Guru was the expert with whom we learned the cryo SCM and developed, and the Andrea was very helpful also with that, who is our technician.
Thank you for your work. If you have questions, maybe you can ask the camera friend that we are late, but if you have some questions, I can shortly ask, and we can discuss that. Yes, another colleague.
If I can ask, we can discuss that. I think talk already about, but I'm not really sure if I really understood if the three different glycocalyx that you presented.
Sorry, it's really difficult to get a company. I don't know.
The question is, are the three different glycocalyx
Yes.
specifically present on the surface of specific cells? Are they present on the same cells? How quickly can it change so that we have modularity between the interaction of the cells?
Excellent point. We actually don't know, but whatever cells we get after the dissociation, we may not get 100%, then we will feel type cells. The epithelial cells, I don't know if we get. We get several cell types within the matrix, and then the matrix is actually should be with the glycocalyx. But we still did not really show that. We did a cryo SCM, I didn't show you that. You can see the lamellar structures also in the sponge, but and this is practically that, or it should be there. We need to identify the monoclonal antibodies. So all the internal cells will have the glycocalyx, and it will be major molecule will be there. So this is but when we dissociate cells, we can see three different cell types, but not the pinacocytes and not the color cells, right? So these are chromocytes, and pinacocytes. I don't know if we have them because when we dissociate, these are they have other dead junctions and tight junctions.
But when you're able to recognize the cell type, do you see only one of the three, or do you see a mixture? If you're able to recognize the cell type,
we could recognize the cell type.
but then do you see if there's only one glycocalyx, or do you see all three?
No, the three glycocalyx from three different species. Yeah, so sorry. No, these are what I showed you in the beginning, yes.
Okay.
But the other question is also whether